Good day, students. Welcome to the lecture recording on the module Food Proteins. This recording is actually aimed at the subject Food Chemistry 2. My name is Ms. Lusane Bangani and I'm going to be the facilitator for this module. Before going into the actual content, what I'm actually presenting to you is the lesson plan. A lesson plan is very important in terms of guiding myself as the facilitator and you as the student as to see where the chapter is going and what is actually required of both the lecturer and the student. So first of all, we talk about the rationale. Why is it important that we have to do this certain section of food chemistry? And then very important, the content. What are the things that we are going to focus on and what are we going to go through in order for you to achieve the learning outcomes that are actually on the left-hand side? So your content has to speak with the learning outcomes, and also at the same time, we also do some certain teaching and learning activities like your puzzles, your video demonstration, word scribbling, and those actually help you in terms of understanding whether you will actually be able to achieve those learning outcomes. Another important feature of a lesson plan will be the references. As you already know that a PowerPoint presentation is usually a summary of work that has been obtained from a certain source. So for deeper and meaningful understanding, you have to visit those references. The most important one that I love the most will be The Chemistry of Food by Jan Velisek. So it is actually available at the library. I also use some YouTube videos and Google and so on. And lastly, in terms of the assessment, so how we're going to assess whether you understand the work, we're going to do some online test on Blackboard and some class exercise, which are actually embedded within the actual PowerPoint presentation. And lastly, in terms of the summative assessment, you're going to do a test 2, and then the FISA. Okay, so now in terms of the chapter outline, what is it that we're going to focus on? We've already mentioned it in the content. The first thing is introduction as to what proteins are. Then we're going to go to the protein structure, whereby we're going to do the simple one and categorize the amino acids based on their functionality. From there, we talk about the nutrition with reference to the type of amino acids that we find, and then we delve in deeper to the levels of protein. Thirdly, this section is going to include how proteins are actually processed and how some of the processing techniques that we apply in food science change the nature of the proteins as to whether it's a positive thing or a negative thing. So that's when we're actually going to talk about those food functional properties, especially the positive effect of processing. Okay, so now in terms of the actual content, what are proteins? I mean, we always talk about proteins, high in proteins, low in protein. I'm taking a protein shake or protein supplement. So what are these proteins? Proteins are actually monomers of compounds known as amino acids that are joined together by what we call a peptide bond, then to form this polymer called proteins. So you have monomers joining together in a peptide bond to form your polymer. And then we actually have around about 20 different amino acids. So those are the amino acids that will be joined together to form this protein. So what are the importance of these proteins? Proteins actually participate in virtually every biological process that is known in biology. So biological process would be digestion, will be growing of plants, anything. So some of the biological functions of protein, I'm going to highlight those that are more or less food related. Okay, so what happens is that proteins are catalytic proteins. Like we already know that proteins are enzymes. Okay, so enzymes like your amylase that's required to break your starch, and lipases that break down your fats during digestion, and those ones are known obviously as enzymes. So they are also proteins, they're very important. And then we talk about the contractile proteins like your actin and myosin, which you're going to learn a lot about when we talk about meat and meat processing and all that. Think about your hormones. We don't study hormones, but one of the most important regulatory hormones like your insulin that actually controls the blood sugar level, that is also a protein. So you can already see that most of these proteins, or all the proteins, are actually important in terms of these biological functions. And then the last one that I'm going to talk about will be the storage protein, especially the casein. We all know casein is the chief protein in dairy products, in milk. Gluten that we get as part of the network in bread making in the dough. So those are the very important biological processes. So in order for proteins to actually perform these biological functions, they have to be obtained from somewhere, and it's definitely from the food. That is where we get in. So this is where we concentrate in terms of the proteins. So the food functions, the first one obviously will be nutrition. For you to be able to take in the nutrients so that it can be able to perform those biological functions that I mentioned in the previous slide. So first of all, in terms of nutrition, your proteins supply what we call essential amino acids. Essential amino acids are what we call indispensable amino acids because the body itself cannot produce via those biological processes. Then we have to get it from the diet. And what would these proteins do, whether essential or not? They in the body where they do cell synthesis and repair, and obviously all the other biological functions that I mentioned there. Besides nutrition, proteins provide what we call satiety. Satiety is actually feeling fuller for longer. So by eating proteins, they are slow to be digested. So you can actually have maybe a 1 meal serving of a high protein food and you can stay full for the whole day, unlike maybe eating white bread whereby you get full and then in a few hours then you are already hungry. So it provides satiety. And very important in terms of us as food manufacturers, it has what we call, it imparts functional properties, and some of the functional properties will be texture. Think about your meat, the different meaty textures, the different flavors in terms of the umami and the Maillard reaction that we're going to talk about later. And very important, they act as emulsifiers. Think about when you use egg yolk in your practical in Food Tech 1, whereby you use egg yolk as an emulsifier when you are forming mayonnaise. So those are the functional properties. Not only that, there are many others, but we're going to talk about those later. And protein aid for digestion. They aid digestion. Like we already mentioned, that proteins are enzymes, and enzymes are there to break down the macromolecules into smaller monomers so that they can be absorbed. However, as much as we've mentioned all the good things about the so-called proteins, we also realize that proteins also bring about adverse functionality, whereby some proteins are allergens, like your soy, your casein. You already know about that in terms of your labeling legislation. And they also provide what we call anti-nutritional factors, which we have what we call trypsin inhibitors. Trypsin is an enzyme that breaks down protein, but when you have other proteins that are named called trypsin inhibitors, they actually inhibit trypsin from working. Okay, so now that we've spoken about the introduction, let's talk about the structure of an amino acid. So the structure of an amino acid, why do we have to say structure of an amino acid and not structure of a protein? Obviously, like I already mentioned, amino acids are building blocks of protein. So before we actually talk about the holistic protein structure, we have to talk about the simple protein amino acid structure. So each and every amino acid contains what we call an alpha carbon. So an alpha carbon that is usually attached to an amino group on the left hand side there, and it's another one is attached to the carboxyl group, and then the other one we have what we call the common hydrogen, and then the last attachment will be what we call the R group. The R group is unique for each and every different amino acid. The R group is the face of the amino acid. Like I had mentioned earlier on, that there's 20 different amino acids. The way we differentiate those amino acids is by looking at the R group. So let's talk more about that. Okay, so in terms of the amino acid structure, there is some form of terms that we need to know, that we need to understand before going on to the actual deep structure. So what happens is that most amino acids are stereoisomers, or what we call enantiomers. We already know that from first year chemistry that these are compounds with the same molecular formula but differ in arrangement. Why? Because we have what we call the L and the D form in terms of amino acids. The L form differs from the D because on the L form amino acid, the nitrogen or the amino group is on the left hand side. Meanwhile, on the D form, the nitrogen base or the amino group is actually on the right hand side. So those two become stereoisomers of each other. Okay, so another thing to note, very important, is that all amino acids are symmetric, or we can say they are chiral. Chiral means that the carbon, the alpha carbon, is actually attached to 4 different groups. However, with glycine is different because glycine only contains 3 different groups. So let's just make an example in the next slides. Okay, we'll talk about the example in the next slides as we go on. But yes, so glycine has a hydrogen as a side chain. That is why it's called a chiral. Okay, so classification of these amino acids. So how do we then classify? We said that amino acids, all amino acids are the same. They are differentiated by the R group. So then we can actually categorize them in groups based on those R groups. So first of all, here in the middle, we have a common amino acid structure. So all amino acids will have a hydrogen, same carboxyl group, same amino group. However, the R group is different. So we have 4 different types of arrangements or categories of your side groups. So we have what we call the positively charged. You can see it with the positive charge on the amino group. You have the negatively charged, the ionized ones. You have the polar uncharged, and then you have the hydrophobic. To just repeat that we have 3 hydrophilic types of amino acids, which is the positively charged, the negatively charged, and the polar uncharged. And then we have the hydrophobic one. So most of the times the hydrophobic one is actually shown by the methyl group. Okay, if not the methyl group, it's shown by the benzene structure. So all these amino acids here are hydrophobic amino acids. You need to know them and be able to differentiate one from the other. I wouldn't expect you to be able to draw them, but to actually identify which one is it. And then secondly, we have the positively charged amino acid. All these amino acids actually have their names. Like there's alanine, isoleucine, leucine, methionine, and so forth. Then we have the positively charged, which there's only 3 of those. It's lysine, arginine, and histidine. And then we have the hydrophilic amino acids, the polar uncharged, where we have serine. You can see it's characterized by a hydroxyl group, threonine by a hydroxyl group. The cysteine is characterized by the sulfur, and in that way it makes it hydrophilic, but they are not charged. And then we then have the negatively charged amino acids, whereby you can see there's a negative charge at the end. So that will then be the categories of the amino acid. This is just a table that is actually summarizing all of those different types of amino acids. Very important, something that I forgot to mention here would be the essential and non-essential part of the amino acid. So in terms of that, when we talk about essential amino acids, we talk about amino acids that the body cannot produce, or the body produces in smaller quantities, which is not enough for the requirements. So as you can already see, I've summarized some of the essential amino acid here. And then we have, of the essential amino acids, you have leucine, lysine, isovaline, and so forth. And then we also have, so meaning that everything else that is not on the left hand side is non-essential amino acids. However, we also have what we call conditional essential amino acids. Conditional essential amino acids become essential only under certain circumstances, like pregnant mothers, people that are sick, maybe with a chronic disease. Then obviously their body is not strong enough to even produce the non-essential. So at that moment it becomes essential. But after the pregnant mother has delivered the baby and she has recovered, the conditional essential amino acids stop being conditional, then the body can produce. So these essential amino acids, if I repeat, they are also known as indispensable amino acids. That's the essential ones. And then the non-essential ones are what we call dispensable. Okay, so the essential ones you have to get from the diet. So if we say these amino acids are essential and they have to be gotten from the diet, then when we are categorizing the food products that actually contain these essential amino acids, we term those products high quality protein products. So the protein quality is quite high, and the reason being the amount of essential amino acids in that product exceeds that of the non-essential. So protein quality refers to more of the essential amino acids and less of the non-essential amino acids. Then we say the protein quality is high, and then obviously the opposite would be low protein quality. Okay, however, in terms of those essential amino acids, they have to be available for the body to be able to use. And what that means is, like back there we spoke about the L and the D amino acids. Okay, so L amino acids are the natural occurring amino acids. If your amino acid are in D form, that usually happens due to excessive processing that will take place and then changes the L to D form. But your naturally occurring amino acids are in L form, and those are the only type that the body will recognize. When I say the body will recognize, is that during digestion, after digestion, we know that all the nutrients are then absorbed by the body or by the cells. So after digesting a protein that contains D amino acid, the body cannot recognize the amino acid in that form, and in that way it rejects it and it's not absorbed. Okay, so and that is termed bioavailable, biologically available, because the biology is there, obviously the absorption. Okay, so from there we come up with the term protein efficiency ratio. The protein efficiency ratio determines how well a protein supplies essential amino acids. So the essential amino acids are supplied well when the protein or the amino acids are in their native L amino acid form. If it's not there, it means the protein efficiency ratio is then low. So it means that if you have your food product that contains lots and lots of essential amino acids, but that food product has been over processed in such a way that those amino acids are in the damaged form, that food product will not, or the proteins in that food product will not be able to be absorbed by the body. And then when we talk about the type of food products in terms of nutrition, we have what we call the high protein quality products, intermediate, and the low. So the high, as you can see, are your red meat, your fish, soybeans, and eggs. However, those ones are very expensive. You get the intermediate, the legumes, and then the low, which are your wheat and potatoes. And just looking at the wheat and potatoes, that is actually staple diet of most human beings. So it means most of the population, especially here in South Africa, the people are actually consuming low protein quality food. So how do we go then about ensuring that even if the people are consuming the food product that are known to have low quality proteins, where are they getting their protein content? So that is done via fortification, whereby with the wheat bread, where you have a high protein white bread because it's been actually fortified with soybean flour, so that even the people that cannot really afford, then they can afford. And then we also have crossbreeding, like biotechnology, whereby the genes of a flour that produces high proteins is then incorporated in the genes of wheat flour, and then in that way it will be able to produce high protein quality. So this one would be just a summary of the amino acid content of some selected food in selected food. So this is an exercise that you have to do. Okay, so what you do is identify all the essential amino acid, and by identifying the essential amino acids, then you will be able to see or confirm that the beef, egg, and wheat, in terms of where do they fit in terms of the protein quality. Is it high, low, or intermediate? Okay, and then we come to the crux of the matter, where we talk about the actual structure of the proteins. And we said we cannot talk about proteins without bringing in the amino acid. So the protein is actually a polymer that is made up of amino acid monomers that are joined together. So in that way, the peptide bond is a bond that links the amino acids together. And that bond is actually formed between the amide hydrogen, so the hydrogen from the amino group, and then the oxygen from the carbonyl group. So when those two form, they form a peptide bond. And this peptide bond is actually formed via a process known as condensation or polymerization. I mean, we're forming a polymer after all, it definitely has to be polymerization. So polymerization, when that takes place, you then have the water being formed as a byproduct, and that's condensation. This is the same process that also takes place in fatty acids when the ester bond is formed. It also forms in carbohydrates when the glycosidic bond is formed. So what happens is that when the bond is formed, you then start having amino acid residues. And in this, we usually call this a peptide. Okay, so that peptide usually has what we call a direction. The direction means that there's an N terminus and then the C terminus. In each and every protein, depending, it does not matter how many amino acids are joined there, you will always have a free amino group and a free carboxyl group at the end, and those are known as N and C terminus. So once the amino acids are joined into a peptide bond to form whatever polymer, they are actually known as amino acid residue. And that peptide bond is very strong, and it's due to the fact that it's a covalent bond with little rotation around it. So it's very strong, it's not easy to break down, but in food science we find ways of breaking down things, don't we? Okay, so also we can also hydrolyze it, like I already mentioned. So in terms of the amino acids, the most important thing is when the amino acids are being joined together. Those 20 amino acids are being joined together. The most important thing is the sequence. So this one is actually showing you a diagram whereby the amino acids are joining together to form a di and a tripeptide. Look at the water being formed. You are requested or required to know how to draw a protein. When given different types of amino acids, you can join them in whatever form that you are required to do. And then in terms of, we already mentioned that when the proteins or the amino acids join together, they form what we call peptides. And then the peptides that form depends on obviously how many of those chains or how many of those amino acids are joined together. So short chains of amino acids, whereby you have amino acids of less than 50, are known as your peptides. Okay, anything below 50 is known as a peptide, and those are your polypeptide. However, when you have two amino acids joined together, you have a dipeptide, 3 tripeptide, and then 4 tetrapeptide. I mean, that is just basic chemistry. And then when you have at least 2 to 10, so you have at least 2 to 10 amino acids joined together, and in that way, that's when you actually call them oligopeptides. For few, it means few. Think about the oligosaccharides that you guys also do in carbohydrates. And then it's called a polypeptide when it has more than 10 amino acids. Made a mistake there. So when it has greater than 10 amino acids but less than 50, because 50 and above, that's when you call it a protein. Okay, so here's just a little bit of recapping the glossary. Glossary is obviously all your terms. So look at all those terms in there. All these terms that are in that picture are the terms that are actually mentioned during the presentation. So just to recap, you need to know what a polymer is. What is a polymer? There is an ester bond. There is protein efficiency ratio. There is amino acid composition, amino acid profiling, and bioavailability. So you need to be able to know this glossary. So for each and every of this term, I would want you to actually define the term. Cysteine is an amino acid. So go back and go check under which categories it belongs to. Leucine is an amino acid. What categories does it belong to? It's actually an hydrophobic amino acid, but it also happens to be an essential amino acid. So you need to know everything about that type of term. And then lastly, before we go on, then we have the glossary. What I'm expecting you to do, and it will be on Blackboard, is to, of all those 3 terms that are actually clustered together, I expect you to be able to construct a sentence that contains all of those 5 or 4 terms. So each and every picture contains terms. You construct a sentence, a meaningful sentence using those terms. You are not defining the terms, but you are constructing a sentence. And then lastly, of the class exercise that will also be on Blackboard, what I'm expecting from you is you already understand the amino acids. You've already categorized the amino acids in terms of their different side chains. So you can already see here is leucine, it's hydrophobic. Asparagine is hydrophilic, negatively charged. We have an hydrophobic one, and so on. So this exercise, what happens, I'm going to put it on Blackboard, but in this exercise, you are required to actually answer questions based on a mother has a newborn baby. She cannot breastfeed. So when she cannot breastfeed, then you know the mothers obviously have to rely on baby formula. So as a result, what would happen is then you go to the shop and you get product 1 and product 2. So 2 different brands of baby formula. But the mother, who is just a mother, is not a food technologist or a dietitian or anything, does not know which one to choose because of the nutrition profiling. So those nutrition profiling are the amino acid profiles of the protein. So now she then comes to ask you, which one do you think I should buy? Which will be the best in terms of my baby's nutrition? And then you are expected to choose one. And when choosing one, then you need to also give a reason why. Okay? So we are actually done with this recording. Just to recap, we spoke about the introduction as to why proteins are important, because they participate in all biological functions, and then for them to be able to do that, we have to obtain them from the food. And then from there, we spoke about the essential ones and the non-essential ones, the ones that we require from the diet because the body cannot produce. From there, we spoke about the actual protein structure, but the simple protein structure by boiling it down to the amino acid. So we mentioned that there's 20 different amino acids. They are all different from each other. And what differentiates all those protein is definitely the side chains. Those side chains are divided in categories based on whether they're hydrophobic, they are hydrophilic, or negatively charged, or positively charged. So from there, and then we also spoke about how those amino acids join together to form a protein. And then from there, we then had some exercises. So I hope you understand the work well. Listen out for the next recording.